Welcome. I am so excited to be here with these incredibly accomplished panelists to discuss the state of the autologous iPSC cell therapy industry. As we all know, the iPSC industry is emerging and exciting and full of potential. In the auto iPSC cell therapy space specifically, there has been remarkable progress, even just in the past 3 months. So in August, I believe we had 2 new INDs go into effect, both for Parkinson's treatments. One of them by Xiaokui's team at Aspen, so huge congratulations there. And so that actually brings us to 4 INDs in effect for the auto iPSC cell therapy space. The first one being from Kapil Bharti's team at the NIH for age-related macular degeneration, the second being from HeartWorks for congenital heart disease, and the third and fourth being for Parkinson's from Aspen and Orion Cell Therapies. So very timely to be having this discussion. We're seeing a lot of exciting progress. And the way I would like to start is by having each of the panelists introduce themselves, talk a little bit about their program, and why you decided to take this route, autologous and iPSC-based, specifically for your drug product. So we can start with Kevin.
Thanks, Marinna. So, Kevin Demore, and my background is 21 years in pluripotent-derived cell therapy. The vast majority of that was with embryonic stem cells, so allogenic, but recently joined Stemson about a year ago, where we are focused on autologous iPS-derived cell therapy to treat hair loss. And why that needs to be autologous, not allogenic, is obviously that condition would not tolerate any immune suppression. And given that our hairstyles are also different, we think there's a lot of value in the autologous approach to maximize the effect of those regenerated hairs matching the existing hairs for each patient.
Hi, everyone. Xiaokui Zhang. Can everyone hear me? Okay. Good. Good? Great. Yeah, Xiaokui Zhang, Chief Scientific Officer at the Aspen Neuroscience. First of all, thanks so much to the organizer and sponsor for having me here to talk about what we do at the Aspen Neuroscience. And it's a really great pleasure to be on this panel to talk about something we're really passionate about. So why autologous for iPSC-derived dopamine neuron in Parkinson? Maybe I'll talk a little bit about who we are and how we started this journey. So Aspen Neuroscience is an autologous iPSC platform company. The company was born in Dr. Jean Lauren's lab, who's in the audience, Jean. And it was over 2 decades of the stem cell biology integrated with the latest artificial intelligence-driven genomics that formed the foundation. Jean and Dr. Andres Braleo co-founded the company in 2018. So we basically were a company that really focusing on development of the patient-derived, the iPSC platform, and also a lot of the differentiation technology to drive the iPSC cells to different type of the cells, starting with a dopamine neuron for treatment of Parkinson. So what we intended to do is actually for those patient-derived iPSC, differentiated dopamine neuron to replace the lost dopamine neuron. Typically, when the patient was diagnosed with Parkinson, the majority of the dopamine neurons are already lost. So not only that, we intend for dopamine, the iPSC-derived dopamine neuron to replace those lost neurons, and also is actually for the cells to have the ability to persist, to form the neural circuit and the network in order to achieve the transformational therapeutic activity. So that's the reason that we're really focusing on the autologous approach and then thinking about how do we actually further optimize the platform and technology to deliver on that.
Hi, everyone. I'm Kapil Bharti from the National Eye Institute. Our program is to develop an autologous iPS cell-derived retinal pigment epithelium patch technology and bring it to patients with macular degeneration. These patients are quite old. They're in their 80s, late 80s often. they go blind because the photoreceptor cells, which as you all may know are the light sensing cells, die in them, but they die because the retinal pigment epithelium cells die in them. And the idea is to transplant a tissue of RP cells, 8 square millimeter, on a biodiverse patch so that these cells can go in and restore the photoreceptor function and stop degeneration of the of the remaining photoreceptors. Why autologous? Because 2 reasons. One is that, as I said, most of these patients are in their late 80s. Some of the previous studies that have been done with stem cells in these patients have seen the biggest challenge was the side effects related to immunosuppression. So these patients had they could not tolerate long-term immunosuppression, and their comorbidities, diabetes, and even cancer-related comorbidities did go worse with immunosuppression. And the second is, historically, people have tried surgically transplanting an autologous RPE from the same eye, cutting from the periphery of the same eye, translocating that into the center where the vision, most of our vision is. And they saw that the graft did survive in a small number of cases, and there was no immune rejection of the graft. Similar work we've done in our lab with large animals, with pigs, and the work Masai Takahashi has done in Japan. We saw that in animals, in pigs and monkeys, autologous RP cells had a much long-term survival than the allogeneic cells. So we think in the long term, these grafts will survive better. And for patients' quality of life, autologous will really be useful.
Thank you. And so we wanted to start by talking about QC characterization at the iPSC stage. It's something that every autologous iPSC cell therapy developer has to deal with. And it's also a situation where all therapy developers seem to be taking a slightly different approach. Everyone has a unique patient population, which can accommodate different levels of risk. So I wanted to hear from each of you, how are you approaching defining specifications at the iPSC stage, particularly in a field that is early, where the FDA hasn't yet said these are the set standards for characterizing iPSCs? What approaches have you taken?
Well, I guess one of the first things you want to make sure for an autologous product is that the cells do belong to the patient. So identity belonging back to the patients is one of the most important things that regulatory agencies are looking for. Sterility is another thing that they're looking for. Pluripotency is not something that they are hard on. You would want to know that your cells are pluripotent. You would want to know that they're making the relevant product that you want to make it for. But that's not the most important assay. So we have a criteria that we have set for pluripotency markers that they should be above a certain percentage, so we know we have made the right cells. But that's not the most important piece. One of the important pieces, or 2 important pieces, are, in addition to identity, is the loss of reprogramming method that you've used, be it the plasmid or mRNA or any other method that you've used that has a possibility of integrating in the genome. You would want to demonstrate that the cells have lost those. So we have a criteria that we're looking for the loss of reprogramming vectors. And the last bit that is most debated is the genomic stability, which you do by karyotyping. And by looking at, one can do whole genome sequencing. There's no regulatory guidelines what to do for this one. We chose to do whole exome sequencing of the oncogenes only, just to be on the safe side. For several years ago when we started this project, there was some concerns that iPSCs may acquire oncogene mutations, so we wanted to be on the safe side. We do an oncogene exome panel and compare it to the existing NCI Cosmic database. So any of those, if we identify any actionable mutations in any of our iPSC clones, we would remove that clone from further analysis or further manufacturing pipeline. So that's the criteria we have.
Yeah. So a lot of approaches that Kapil just described are very similar that we incorporated into the processing, starting from the fibroblast cells that derive from the biopsy, skin, very small piece of the skin biopsy that obtained from the patient. So maybe what I will elaborate a little bit is actually, so at the very beginning that we do the SNP array, we establish the barcode, molecular barcode for each patient. So then later on, because our manufacturing process is multi stages, so that for each stage, basically we do this assay again to ensure there's no swap or mixed up that during the processes. So then, and also some of the whole genome sequencing approach Kapil just mentioned, those are also part of the what we do. So we basically call that a quality filtering system. So not only, right, we use the whole genome sequencing to ensure there is no new generated or introduced mutations during any stage of the manufacturing, specifically reprogramming, and then later the differentiation to the dopamine neuron. So in process and also at the product release, we perform those whole genome sequencing to very carefully interrogate the genome integrity. And we do use NCI, the database, the portal, where basically the cancer-driven mutations across over, I think, 60 type of the tissues were there. So we regularly update the list that's specific to brain because our application is specifically intra-brain, in the application. So this is how basically we're ensure each stage that iPSC cells, passing all those quality controls. So on top of that, and then years ago when Zhang started kind of think about how to characterize those iPSC cells, and together with some of the other progenitor stem cells, right? He generated this large reference data set in collaboration with many labs globally. So that actually has become a machine learning driven, the predictive tool that we utilize to evaluate the iPSC quality, specifically as a pluripotency. So we do utilize that to evaluate basically all the iPSC clones that derive from the fibroblast, and to be able to select the best clones that going forward.
Just to echo pretty much everything Kapil and Xiaokui talked about, genomic identity, genomic integrity. iPS affords the opportunity to compare the genome of the iPS cell to the actual donor cells. So this is something that's not possible with embryonic stem cells, and it's still true to date, I think, that most of the clinical activity has so far actually occurred with embryonic stem cells, but I think there's an emerging theme among the regulators that you absolutely need to be comparing the starting material to your iPS cell and probably also your cell product material to evaluate what may have changed in the genome over those sometimes very long sets of processes of reprogramming and directed differentiation. Going beyond, say, pluripotency quality control assessment as a functional attribute of the cells, I think it's important to measure the purity of the starting iPS cells prior to directing their differentiation. In many cases, if you have spontaneous differentiation during pluripotent culture, it is likely not generating target cells for you. So it's much harder to contend with that after the fact than before. So we think the purity of pluripotent cells can often be best assessed by looking for things that you don't want to be there. So markers of spontaneous differentiation, I think, is an important QC step for iPS cells. When it comes to genomic integrity, certainly whole genome and/or whole exome sequencing is very important. The tools are out there to do it. I think the tools are just emerging on how to interpret it. And that's probably going to be the biggest evolution in this field going forward, is what we do with that mutational spectrum information, because it's always going to have mutations, right? It's just is there an impact to them that we can ascribe? And then a little bit harder than that sort of small mutational spectrum is looking for structural rearrangements that aren't the easiest things to capture with short read sequencing. There are a variety of technologies out there, some of them fairly new and emerging, but I think they're going to play a major role as a QC, a set of QC steps for iPS cells overall, and even end product cells, provided they fit the bill for sort of a molecular characterization. Many of the cell product cells we might deal with aren't necessarily proliferative enough to use, say, G banding. So that's where I think molecular tools for genome characterization going to become essential or already are essential.
Great, thank you. Following up on the characterization thread, there was a conference that a few of us attended last week where Century Therapeutics gave a really nice presentation on how they were able to use a series of almost one-time experiments to demonstrate that there was a very low risk of residual gene editing plasmid left over by the end of the drug product manufacturing process. And what I thought was so powerful about that set of experiments is that they were able to use it to make a case against having to do more extensive, burdensome, batch-by-batch characterization of residual gene editing plasmid for each of their drug product batches. And so given that one of the things that is necessary for autologous iPSC cell therapy manufacturing is that you have to manufacture and characterize for every single patient that requires a therapy, wanted to ask if there are any cases in your drug product development where you've seen it's possible to take this sort of approach, where you can do a series of clever one-time experiments or any other strategy to kind of reduce the burden of batch-by-batch characterization. So I'm going to start with Kapil.
So I think there are multiple ways one can address this. So the concerns for autologous is, in addition to the leftover gene editing products, what about leftover iPSCs? So one has to address that as well. In our case, for instance, it's a polygenic disease that we're treating, so we don't have to gene edit the cells, but we still have to demonstrate that there are no iPS cells left. So we actually had a similar experience at Century Therapeutics, where we did a series of experiments where we actually deliberately spiked our RP cells with iPS cells at different gradation, different percentage, and let the cells mature on the final scaffold that we use for transplantation, and demonstrated by multiple assays, flow cytometry and qPCR and whatnot, that the iPS cells cannot even survive the final media that is used for maturation of RP cells, and use that as a basis to design the preclinical studies that we don't have to do spiking studies in animal models, and also to show that we don't expect any iPS cells left in any of the process, any patients, every batch that we make from patient to patient. So that's one way to look at it. The other thing that we implemented in our process is an AI-based imaging of the drug product. So our RP cells, like I said, they are on an 8 millimeter tissue on a scaffold. There are about 75,000 cells on the scaffold. We developed an AI algorithm that was able to recognize every single cell and convert it into a hexagon, because these cells are hexagonal. And that once that segmentation was correctly done, then we had complete shape matrix of all the 75,000 cells. So every cell that is going into patients, we know what it looks like. And comparing that to what RP cells would look like or what iPS cells would look like, we could say by shape analysis also, we don't have any rogue cells in our final drug product. So there are multiple ways to do this. And I think everyone should try to find what's going to be specific for their product, what assay they could design for the drug product to demonstrate the complete loss of iPSCs, because that's going to be not a starter that if you have any iPS cells left in your final product. You do want to demonstrate that you don't have any. And also that provides, in addition, kind of a QC assay for the quality of drug product that we have made, that we have pure RPE cells in our final product.
Yeah, sure. So I agree. This approach actually can work very efficiently in terms of what specific models or assays is probably indication to indication, or cell type to cell type. So in Aspen Neuroscience case that we have conducted 9 months, the GLP, the study to evaluate our product that are referred as AMPD-001 in the new rat model, which we presented the data in the summer at the World Parkinson Summit. So basically in this study, there are actually specific objectives that build into this one study. First is actually we conducted this transplantation of AMPD-001 intrastriatally. So then we'll be able to evaluate any potential tumor genetic activity, if there's any. And also we're able to evaluate the biodistribution. If they actually work like a neuron or persist as a neuron, we should be able to monitor that through the 9 months period of the time. And the other is that just morphogenetically, right? And also through the staining and see if the cells actually proliferate. So basically, the results that demonstrated there's no residual iPSC cells that detected. So we actually used a similar approach. We spiked the iPSC into the drug product. So from the control, you were able to see the teratoma formation, but in the drug product treated the arms, there's no teratoma or any concern towards the iPSC, the presence. And also we're able to see the cells actually, they're where they are. They didn't migrate beyond where the cells are introduced. And also the cells actually, since they're post-mitotic, so that we didn't really see the proliferation in those arms. So then maybe I'll kind of talk a little bit about the Neurotest, which is another RNA-seq and the machine learning based genomic tool we utilize for each product release. So basically what this genomic approach do is to predict the maturation of the cells. So basically they're at a very specific stage of the differentiation where they still have the ability to form the circuits and the network, but they're post-mitotic. So that can significantly de-risk for some potential tumorigenic effects. So it is a combination of the multiple approaches, quite similar to what Century has been utilized in their development.
Great. Kevin, any thoughts or strategies around streamlining characterization?
I think the biggest ones have already been mentioned by Kapil and Xiaokui. It's sort of in vivo safety testing, which obviously would not be practical on a patient by patient by patient every lot. I think reprogramming reagents, that's probably an area, like we don't want to necessarily passage iPS cells for 15 passages just so that we're sure there's no more Sendai virus or whatever left over. So there could be a role for really studying analytically when those reagents are gone and being able to trim down your passage numbers before you have a suitable starting bank of iPS cells. Coming back to residual iPS cells, I think it's a problem for the field overall. I know I've dealt with it in past settings as well. The best we can do is spiking, but to be honest, a pluripotent cell that is residual after being exposed to a differentiation process is likely not the same as a spiked iPS cell. So I think there still is a role for molecular analyses, whether they be qPCR or protein-based, ddPCR, to look for residual marker expression that's specific to pluripotent stem cells, just at least at this relatively early stage of evolution and development of this field, because I don't think we can necessarily rely on naive iPS cells not sticking around in some sort of condition to be the same as something that escapes differentiation in the first place. And hopefully all the deep genomic characterization helps assure that.
There's another point I want to make here, which is, I think, relevant for autologous, especially autologous iPSC therapies, is that why this test would work is because for autologous, process is a product. So once you've validated your process with these kind of analysis a couple of times as part of the IND-enabling studies, you don't have to keep repeating it for every patient as part of once the trial has started. Unlike for an allogeneic, where every time you make a bank of your cells, you will have to go through the complete testing of the bank because your product is a final drug product. So I think that's an important distinction.
That's a really good point. So digging deeper into process development, Kevin, you mentioned passage number. I know we had a prep call where the topic of passage number came up, and how do you decide when to start the differentiation process, at which passage number, what range of passage numbers can you accommodate, keeping in mind that one stem cell scientist's passage 10 is not necessarily fully equivalent to another stem cell scientist's passage 10. So this time, let's start with Kevin.
Yeah, I think everyone acknowledges the manufacturing time commitment and, therefore cost for autologous therapies is probably single biggest hands down obstacle to overcome or issue to address. So I think keeping these passage numbers as short as possible to help reduce total time for manufacturing is important. It really comes down to population doublings. I know that's not necessarily what we're measuring when we count passages, and different culture systems will have different amounts of population doublings at set numbers of quote-unquote passages. So I think we ought to try to refine our techniques to try to measure population doublings as best we can. Maybe imaging is a great tool there to really follow it, as opposed to just cell count seeded and cell counts taken off the plate at the end of a growth period times however many passages. So really trying to characterize, and it might not be the same for every reprogramming technology, I'm sure it's not, how long you need to expand those to lock in a pluripotent, functionality and a truly usable and stable iPS cell bank and clone.
Yeah, I think on top of that, some of the imaging-based analysis that can provide that additional information. And I think at the end of the day, it's all come to, how do we standardize it, right? The passaging, or determine at what specific passage, the cells actually will go to the differentiation. So I think right now we're still in the middle, generating a lot of data, imaging data. Of course, you mentioned about some of the pluritest related genome-wide characterization. We're also trying to analyze the SPECT media. So basically, we'll collect a lot of information, try to understand the efficiency of this very specific unit operation in the entire end-to-end kind of the manufacturing processes. So in the future, I think again, listening to the first session, you were talking about the digitalization. I feel some of those kind of approaches will probably be integrated that will be able to make a decision, right, based on all the data generated during the passaging or proliferation, and help us to really be able to generate a consistent that kind of quality and standardize the procedure for every patient and every clone.
We're big fans of image-based analysis at Cellino, so I love that this thread is coming through. Kapil, how did your team approach things like deciding which passage number to start differentiation?
Yeah, I guess I would say there are 4 competing interests when deciding what passage to use of iPS cells. You do want to make sure that your cells have lost the reprogramming vector, whatever it was used, that they're pluripotent enough to make your final product. But you don't want to go too far out that they accumulate too many mutations or your cost of manufacturing keeps increasing unnecessarily. So you want to find that sweet spot when you say, yes, the cells are ready, they've lost the reprogramming plasmid or vector, they are ready for differentiation, but it's also not, I'm not spending too much time in clean room. So that means early as possible, and whatever that sweet spot is for every drug product. I'll give you an example how we came about it. When we were preparing for our IND-enabling studies, multiple operators were doing passaging and reprogramming. And we saw when the cells were sent out at a given passage for testing for if they've lost a reprogramming vector, there was one operator that were consistently failing more than others were. So we went back to the books and see what was the difference. And that operator was consistently, going back to your point, splitting the cells half a day early, right? So saying that it's confluent versus not, is in the morning versus in the afternoon. And overall, on the average, there were total number of days the cell stayed in culture was 2 days lower than other people who had consistently passed. So that was our threshold then. Then we said we want our cells to be in culture for this many days minimum before we say they're ready for transplant or ready for differentiation or QC.
Yeah.
So that's how we came up with minimal passage number.
Thank you. It's very interesting. Okay, so switching to more general questions. This field is in its nascency. You are the pioneers. What advice would you give to folks who are developing auto iPSC cell therapies that might be in an earlier stage than you? Or another way of phrasing that question, what lessons were particularly hard learned? What things do you wish you knew when you had started?
Every lesson was hard.
Everything is painful.
Retrospectively, you're like, oh my God, I could have done this differently every step of the process. I think it's important because our starting point often is our drug product, because we know how to make a specific drug product very well. So I think focusing on that making a process, a differentiation process from iPS to drug product, make it as forgiving as you can of the quality of the cells you're starting with. And that would really pay off in the long run, especially for autologous manufacturing. Because if you have a way of cleaning up the variability in differentiation, you're going to get variability in differentiation. Everyone in this room is different. Our genetics is different. Our epigenome is different. Our iPS cells are going to be different. There will be huge variability. But if your differentiation process can be forgiving, that will help. And the second piece, if I can tell right now, based on our current experience as we're in the clinic, is start preparing your clinical sites as soon as you can. And I know Aspen has been ahead of the curve for that. Really kudos to that idea. Really start working on that, making sure that not only your surgeons know what to do, your patient coordinators and nurses know what to do. Just to give you an idea, at the early stage of the trial, when you're looking for really late-stage patients, sometimes we're screening 100 patients to find one patient that we will enroll in the study. So for nurses and the patient coordinators to say the right words to them, that this is a phase 1 safety feasibility study, we don't expect that your vision will regain from the study, but yet we would like you to participate because there's a hope for all of us, hope for the community. So with that altruistic reason, you should participate. Giving that message, that training itself is so important. So every bit of it. So, just to give you an idea of, everybody knows electric cars right now. You don't want to just keep making the batteries. You want to make the battery with the entire car and put it on the road as well to see how the car will run in a real life scenario. So I think every bit of it, working on it from early on, is going to be important. It can be overwhelming. It will be overwhelming, but I don't think there's an easy way out. We have to get those products to patients as soon as possible.
Yeah, that's really a great lesson and advice. So again, in this area, I feel a lot of folks have contributed so much that when it comes to analytical, the systems, and this is the area I feel we and together with other leaders in this field will probably all come together and say invest that the analytical development as early as possible and as robust as possible, because you actually will see a lot of variabilities that from very beginning, right? And the patient-to-patient material related variabilities, and then once you start putting all the different unit operations together, each operator will have that kind of variability. Every day, right? Temperature changes, the CO2 running out, and this and that adds additional complexity to the data that we generated. So without that robust a battery of the assays, it's going to be very challenging to dissect which parameter contributed to the variability. So what that will hinder is that you just won't be able to make a decision on a process that you can log, and then start generating the clinical equivalent or clinical grade material for further development. So definitely this is an area that you really adhere to the biology, adhere to the science that you're developing, and then really identify the assays where the mechanism of action, ideally to be reflected. If it takes some time to get us there, then generate a battery of the assays where you can rely on for every batch and every patient-derived material characterization.
So a couple of the lessons hard learned, perhaps for me, actually come from the allogenic field, but I think they apply to autologous. Extending on what Kapil's saying is that the product is the process here, so I think they're in some ways even more poignant for autologous iPS-derived therapy. And one is the directed differentiation. There's almost no way to get too familiar in too much data and too much process exploration around directed differentiation, right? When you need that process to work the same across all the both genetic and epigenetic variability that will come forward with autologous iPS products, you really need to know every single factor you add. You need to know what it does when it's 50% more and 50% less, if not 3 and 10 times more and 3 and 10 times less. You need to know the cell densities, what are the ranges around that at every single programmable input step. How does the cell density affect it? And then there's just environmental things and, some things that learned the hard way in the autologous or allo, where scale up is the challenge. Like when you move from incubators with dynamic pH and static gas to bioreactors with exactly the opposite, static pH and dynamic gas, that's a fundamental change to environment. You can use exactly the same media and the exactly same reagents, and you don't necessarily get the exact same results. So there's almost no way you can learn too much about the directed differentiation process. The other is more the animal models. It's similar, but really understand your animal models, why they're working when they're working, why they're not when they're not, because that's just something that while we're not going to use animals to release autologous product on every batch, really knowing upfront, the sticky points of a given model are really important to be able to interpret that variability that we all see in the animal model outcomes.
Yeah, you mentioned challenges in scaling up manufacturing with allo. One of the oft-noted challenges of taking an autologous approach is scaling out manufacturing, just the complexity and even just logistical challenges of having to do patient-specific manufacturing. And the field is early now. We're not yet in phase 2Bs of clinical trials or commercial scale-up. But how are you and your teams thinking about this challenge and looking ahead and planning for the future?
Yeah, so the scale out for autologous, I think, fundamentally different challenge than scaling up, but I think, industries, multiple, have dealt with both facets in creative ways, so I think we certainly can deal with it. Automation is one that primarily comes to mind for the scale out, but with automation comes miniaturization, right? You don't want, monster robots that are mimicking what humans do. You want to sort of re-envision, like, here's the process step. How do I make something that does that more automated? Maybe it's not fully automated in a Gen 1, but it's partly automated, but miniaturized so that it can be parallelized. That's the key. I think there's even some regulatory frameworks that need to change, like line clearances and what you do in a clean room or how you design a clean room. I think there's some things that need to evolve in that front for scale out of autologous therapies.
Yeah, I agree. I think for autologous cell therapy, the scale out, a lot of the patient segregation strategy and all that are the area. I think right now we're so early. We just started kind of scratch the surface. The autologous CAR T paved the way. However, a lot of the miniature type of cocoon or other type of the vessels are not going to work for neurons, right? So this is an area that I think this kind of ecosystem, right? The technology developer, technology adopter, and then therapeutic developers really need to work together, to come up with the solutions. And the Cellino right now is leading, one of the solutions that for autologous cell therapy. Definitely that we welcome more considerations and strategic type of the planning together across the industry to get us there. We don't really need billions or trillions of the cells, right? But we do need the capacity where we will be able to scale out for many, many patients. So we don't have to kind of wait and then wait for one batch to be completed and then you go to another. The runs, the clinical campaign needs to be effective as how many patients the patient population needs. But this is definitely, we're so early. So we are, again, Aspen's among many other therapeutic developers actively looking for this type of the manufacturing partnership, and then welcome the technology developers then to come to us, and we strategize around this challenge together.
So what I'll add there is, I think the two biggest challenges, and I think both Kevin and Xiaokui pointed towards that, is for logistics is reagents and workforce for scale out. Reagents, especially because it's a long manufacturing process, you want to make sure that you have all the reagents. You have done a risk assessment for all your reagents, and you have identified the weak points for the reagents that where the vendors may be interrupting the supply, making sure you have agreements with the vendors, maybe changing the name even where you need to requalify a backup reagent. All of that needs to be done ahead of time, just so that everyone knows even a change in name or catalog number will disqualify a reagent from the manufacturing, may disqualify, even though the ingredients will not change. So all of this should be done in parallel. These are the minor details that as you get to the process, and for scale out, this will immediately impact the whole scale out operation. Workforce training, if it's a 4-6 month long manufacturing process and people are turning over, you don't want people to be leaving in the middle of the manufacturing process. You want to have, if they're leaving, you would want to have backup people ready to take on the process. Having them trained on such a long manufacturing process will itself take time. So all of those are challenges, and as Xiaokui pointed out, automation, as you guys are leading, is probably one of the ways to solve all of those scale-out challenges.
Okay, so one last question before we turn it over to audience Q&A. If you could wave a magic wand and have any wish granted, any scientific challenge overcome, any technology is developed and works perfectly every time, or any change to the existing regulatory framework, what would you wish for? Maybe we start with Xiaokui this time, because we don't normally start with you.
Are you sure you want to give the magic wand to the
You got a
scientist?
magic wand. You can have 2 wishes.
Yeah, there are so many things that I wish that kind of now looking back that we would have a different way to do it, to accelerate it. And again, we did a lot of lesson learned and debriefing after our IND being cleared. I think one of the areas that we're kind of really thinking that, and Nicole actually, Verdun just talked about it, right, which is a partnership with the agency. How do we strengthen that? How do we actually, right now we're part of the ARM, ARM is a really great sector advocate, really leading a lot of scientific changes. And how do we actually utilize this type of the ecosystem that to ensure a really frequent and active exchange on the scientific development, especially when it comes to new technology that we're introducing to the therapeutic development. I think that could really help speed up some of the early development work. And the other area, and I'm glad that Kapil touched upon the raw material. I think this is an area, especially that during the early stage of research, right, in the bench, I look at the 10 items, right, and then if that works and it's cheap, I'm going to switch to it, right? So that kind of a mentality, development mentality, you're looking at the process and think about how the process changes the impact on the product. So a lot of this is really the CQA type of the, really understand the critical quality attributes as early as possible. And then establish a really robust analytical tools and help us establish it, and then pressure test it through the different runs and different characterizations. That can save us a lot of efforts as well. Otherwise, you're just going in circles. You keep going back to where you started and then wondering where that run or efforts will lead you to. I'll pause and then let Kapil and Kevin address that.
Go ahead.
I think I would use my magic wand to handle line-to-line variability of the differentiation process. Again, spending most of my career with embryonic stem cells, I'm curious about epigenetics, epigenetic memory among different iPS lines as being a contributor here, and like to optimistically think maybe something like the recently reported transient naive reprogramming to sort of further reset the epigenetics of an iPS cell might be something to be explored as in this avenue to improve subsequent reproducibility of differentiation.
I think that both of you said it right. I would say really improving the process robustness and ensuring that you can scale it out, and that's the key. The process robustness is a key to scaling it out, is probably the main thing that any magic wand, for which a magic wand would be needed. But then having, we all who are in the autologous space, if you are doing the calculations for how many GMP rooms and how many workforce you will be needing as you reach phase 2B or beyond, you do realize that the amount of resources and infrastructure needed is going to be immense, and that's where probably a magic wand is needed as well, either making it automating or having, I'm looking at Uma, having a 30,000 square foot facility available to you. So some of those are challenges that, as Xiaokui pointed out, with new technologies coming up, would be solved. Another piece, again, I'll go back to patients, having patients be available, be there, be willing to volunteer for these kind of trials is not a challenge that we should ignore early on, because, and really working with non-patient, non-profit organizations and patient groups, ensuring that we educate the patients what they're signing up for, and also educating them on difference between the snake oil stem cell therapies versus these true stem cell derived cell therapies. So that, I think those are important things, and we have to be kind of working on all of those things in parallel.
Great answers. Okay, so we have time for a few questions from the audience. We have one in the back.
So I do applaud the notion that we need to monitor population doubling time or population doublings versus passage times. And the reason being, Grid's also published some work. I'm loud, you guys. Can everybody hear me?
We can hear you.
Okay. So this is heavy, so I can't talk long. And what he showed was that while there is telomerase activation with the Yamanaka factor induction and reprogramming, it's quite variable. And the efficiency of telomeric elongation is quite variable in your iPSC products. Being vigilant to that might be important, more so on the allogeneic side where you want to do large expansions to make a lot of product. At some point, you might want to double check either the source material to see if they inherently already have a basal level of telomere length that's pretty good, and you're tacking on more T2AG3 repeats to make sure that you have a durable product in vivo in the clinic, but also to monitor that you're not depleting and have cells, because when you get to short telomeres, the cells are going to look fine and act fine in vitro in a short-term assay. But when you go in vivo with them, they may become exhausted a lot sooner, and you lose efficacy a lot sooner, which leads to variability in the clinic. So I applaud the notion of monitoring population doublings in that context.
That's more of a comment.
Yep.
Yeah, well, thank you.
Yes, thank you.
Any questions? See, oh, hey. A lot of friends in the audience.
Thank you, Eugene from Brain Cell. Hi. My question is, I guess, a couple parts, but I mean, one of the advantages, probably everyone here who's a fan of autologous products realizes this is sort of the ultimate personalized medicine, so that's a distinct advantage, but then it's also a knock on this approach, which is, and you mentioned line-to-line variability, do you have a process that's sufficiently robust to work with all the various types of different donors that might come to you? So I guess my question is about your sort of real-world experience in terms of what is the donor-to-donor variability that you're seeing in each of your areas, and is it a real issue, or is it a sort of perceived issue that we should dispel from the field? And then another sort of related question is, if you see variability, what's tolerable, like in your target product profile? Is it okay, our process works for 9 out of 10 people, that's what we're going to go with, or do you need 98 out of 100, or sort of what's your cutoff, and what are the variables that are making you, forcing you to make a cutoff? I guess that's 3 questions.
So I would say the variability is there, and definitely from donor to donor there is variability, but at the same time that variability is tied with the subjectivity with which an operator is handling those cells. So you cannot really completely untangle those at this point. Nobody has done those systematic experiments making my cells with 5 different operators and see how variable those are. I don't think anyone has done that. But be as it may, we can, and since we can untangle those, so we'll have to live with that. So we just call it variability from donor to donor at this point. But it's manageable, and like I said earlier, if your differentiation process is forgiving, that means if your process is robust enough that even though your pluripotency cell quality was slightly lower from one person to another person, your cells, your differentiation process will be still good enough that you get the same drug product or similar drug product from both of them. I think that is doable. In our experience with our process, I would say by now we have tested it over 100 different donors. Not all in GMP, but I would say at least in GMP, at least 18 different donors, and for each donor, at least 3 to 6 clones. So we have significant experience in this space, and our differentiation process is over 95% time successful. So that 5% that it fails is likely because we ended up picking up really a bad iPSC clone that failed. That's our interpretation. How do you, and then your second question was that how do you ensure that it will work? I guess, what is the cutoff for variability or acceptance? I think it will be drug product dependent. And I think Xiaokui said that earlier, if you understand the CQAs of your drug product really well, the critical quality attributes, you can say anything above this is a good product. And you have done enough work during your IND-enabling studies to know that your variability is above this range, so not below that range. So that of passing, and everything above that is good enough, or is what the product would work. Good enough is not the word we're going to use here. So that's the way to do it. That's the way we did it, and that is combined with a lot of in vitro testing of your product, understanding of the science and the biology of your product, and doing animal studies.
Yeah, maybe I'll add. Eugene, thank you for the question. So I don't think I have the answer, but I kind of share with you how we're brainstorm on this variability. Again, we're very early, right? I mean, we have not kind of started to see massive clinical data, so a lot of variability is still to be correlated that in the future setting with some of the clinical outcome. But how Aspen by the way, Aspen, founded by Zhang, Andres, and with a lot of support from the Parkinson patients. So one of the ways that they supported the program is actually donate their cells. So actually through that generated some kind of experience and looking at the donor to donor, the variability, as Kapil already highlighted, that exists at the deriving the fibroblasts in terms of the yield and the proliferation rate. There is a donor to donor variability, reprogramming efficiency, right, and then the differentiation. However, I think it's through the years, and especially some of the predictive genome tools, that we learned how to actually be able to select clones, iPSC clones, or as we differentiate the dopamine neurons, and we basically build into the redundancy, so that we ensure for each patient that we select the best quality of the product. Through all those kind of analytical the procedures. So in the future, what we're hoping, our vision is actually really to be able to establish this integrated predictive tools. So at the very early stage, then through a lot of data that we can capture, right? We can predict how the product, which one to select in process. So you kick out the ones that it's going to fail, and then you adhere to the end what it's going to give the desirable clinical outcome, the product. Or generating the data, we'd be happy to share once we know how to correlate that, how to predict that.
Just simple line-to-line variability definitely does exist. It's not imaginary. It's not a theme that hasn't manifested. What's its impact, I think, is context dependent for each cell product and each differentiation process. Having a lot more experience in endoderm and pancreas, I think Gordon Keller actually did some really good early work just looking at line-to-line-to-line variability, and the same signaling factors are the ones that get used for highly effective differentiation endoderm and or mesoderm, I think he published on. But one line might need 2.5 nanograms of BMP. The other might need 5 nanograms per mil of BMP. The other one might need 7.5, right? So if there was a way to predict that, you can harness what is otherwise intrinsic variability and make a robust process that can handle it. But I do think that remains to be a challenge, certainly for some applications, maybe not everything. And I don't recall exactly your methodology anymore, Kapil, but if you have a process where you can selectively proliferate out a progenitor, I think that is more forgiving between lines than exclusively directing fake commitments among a panoply of potential options using gradients of signaling molecules in vitro.
I think we have time for one more audience question.
Thank you. It's a very interesting session. I just wondering, how do you guys capture the data inside your system? Do you have any specific solution in terms of quality system-based electronic data capturing? Because your product obviously are so complex, you analyze in several attributes for each of your cell line, right, for each patient. So how do you capture, manage this amount of data? Do you have a specific, commercial available solution, or you just develop your own? Because I think data-wise, it's also very sensitive, right? And I guess FDA would also question, especially in commercial phase.
My answer is very straightforward. Build a really strong bioinformatics team, a very strong quality team that at the very beginning, kind of that aside, right? At the very beginning, I think it was actually think through how the data will come together. A lot of us started by data dumping, right? I mean, it's kind of kitchen sink, right? You generate a lot of data, and then when you look at it, you kind of start to lose track. So this kind of goes back to, at the very beginning, what we talked about, right? You kind of really need to have that product in mind as you started to generate this type of the data. Some of the data is to support your process development, understand the robustness, understand the CPP or CQA, right? Some of the data actually, towards some of the in vivo, in vitro, or hypothesis generation. So it kind of really need to sit down and cross-functionally and talk about those strategies, the process, product, R&D enabling, and then in order to actually have the systemic way analyzing and organize it. I'll be happy to kind of share a couple examples offline.
Yeah, we have an online system that was custom built called STEM Labs that we use for data collection. But I think the important bit, as Xiaokui pointed out, is why do you need data collection? Of course, for regulatory reasons, for tracking purposes. But the most important bit for early stage is for analysis. The example that I gave earlier, the only reason we were able to track that population doubling versus passage was one operator versus three operators. Everybody's splitting cells. Everybody's calling their cells at certain passage, same passage. But then one had taken 32 days and others had more days in culture. And we could do that because we went to our data repository and said, pulled out all the data because it was logged on a daily basis. Batch records were logged on a daily basis. So that's the important bit. Early on, that kind of analysis is so important, and you need to have a system in place where you can do to analyze the failed runs. Why did they fail? And then learn from those so you can improve your process iteratively.
Yeah, the data science team that's focused on that, and interfacing with R&D to enforce the principle of collecting that metadata and the importance of it. And early on, getting out of Excel tables managed by R&D staff to, professional data handlers and engineers and architects and bioinformatics folks. So.
Yeah.
Yeah. The idea of having multidisciplinary teams is so important for developing a complex product because not every person can have the expertise that you need to get everything done.
Yeah, so true.
Okay, well, big thank you to our panelists. You're doing incredible, important work, and we appreciate you taking the time to be on this panel.
Thanks.
Yeah, thank you.